There we go. Okay, thank you folks for joining us for our final colloquium of the fall 2025 semester. My name is Craig McCashin. I'm the Honors College here at A&M Texarkana. And the Honors Colloquium is an opportunity for students, faculty, and in some cases former faculty colleagues to present work that they have done. So for our honors students, this is usually either honors projects that they have worked on. For my faculty colleagues, it is usually research in an area that they're kind of investigating. And today we are very happy to have Dr. Ben Neuman, who's professor of biology at Texas A&M University down in College Station. Dr. Neuman used to be the head of biology here. He went down to College Station about 5 years ago, and thus far has not realized that he's actually under no contractual obligation to continue giving this lecture every year, but he does, and I appreciate that. So this is our annual Neuman holiday lecture on viruses and virology, or also called recent developments in research into virology. We are also very happy today to be hosting members of the Texarkana College Honors College. Thank you, folks, for joining us. And yeah, Dr. Neuman, you take it away, and then there will probably be some time for questions at the end. Thank you.
My mother's trying to get in, but she's on Teams, and I don't think we're on.
Oh dear. Well, when she comes to Zoom, we will let her in.
That's fine. All right. Yeah.
Poor mom.
Then let's share things and let's start the show. Oh my goodness. Got so much junk here. There it is. All right. Yeah. All right, so I'm a virus researcher. I grow them. I have people grow them for me more and more these days, and I like finding new viruses. I'm going to show you some stuff. It's almost all stuff that we're doing in the lab right now. There's one thing that's from a friend's lab that I'll tack on at the end, but I don't know. It's just like a little grab bag of everything that's out there because what says the holiday season more than a good case of flu or COVID, right? You know, that's the world we're in now. So. And so, yeah, we'll get a little virus hunting, a little bit of all these other things in there. I want you to end up feeling not too bad about quantum optics. That's part of the goal for today. So let's go. Let's try. Let's try and go. There we go. All right. So one of the things that I do is virus hunting, and we do that with a computer. It's really safe. All you're doing is going through all of the sequences that humans have ever recorded. Because when they're out there sequencing a panda bear or a tree or a leaf or something like that, they'll pull out all the genes from that and accidentally, as like a bycatch, like all the delicious dolphins that get into your tuna, basically, you will get little bits of virus coming in. And we use a computer to scan over all these data sets and find any data sets that have little bits of virus in them. And then we have another computer program that will then do its best to put those bits together. And so we call these virus hunter and virus gatherer. Isn't that cute? Yeah. Now, the problem here for me is that Virus Hunter is awesome at its job. It is absolutely a savant. Virus Gatherer drops a couple of those, whatever they are, berries or twigs or whatever that hairy thing is that it's trying to collect. And so it has some problems building out the little pieces that it's able to find. And so that's where we come in. We've been trying to do this with AI. So far, the AIs are real bad and end up eating up all the memory in all of our computers within a couple of minutes. So just chaos. So we're taking like this broad look at where are coronaviruses in the world. And here's the first page, and you can probably see a theme here. There are tons of coronaviruses in fish. Now, if you're looking at these like I do, you'd say, all right, which of these is the best fish? Best fish, I think, has to go to the leafy sea dragon. That's this one right over here. But if you're talking about fish with the best hairdo, fin do, whatever it means, I think it's got to be this Siganus over there with that little spiky, very punk looking do. And here's the next page, and it's fish all the way down, basically. The best fish on this page, obviously, Synchirapus splendidus. Look at that up there. So there's a ton of brand new coronaviruses. Nobody knows that these things exist except you and me, and I haven't published them yet. So yeah, that's how it's going to stay for another month or two, hopefully. But we find something else in there. Now, first, a little aside about these fish. So the one in the middle, the leopard grouper right here, Plectropomus, is actually one of these strange fish that is able to change biological sex. It'll actually dissolve its sex organs and regrow sex organs of the other sex at a certain point in its life. And it does this thing at the top, which is called protogyny, which means that every single one of these things starts out female and only if they are basically the biggest, baddest female in the bunch do they get to become male at the end. It's like angels, but they don't earn their wings. They earn a pair of something else, basically. So this other fish is also able to do that, and the thing that it earns is this giant spike back there. So that's the Synchirapus fish, and also the grouper. A lot of groupers do this. Yeah. So. What is this weird thing we found in there? Well, it's a coronavirus because that's mostly what I look for. It's what I enjoy. But do you see which coronavirus it is? Yeah, look over there in the subject reference column, and you see those are all SARS coronavirus 2. And then if you look two columns over in the percent identity column, they are 99.7 to 100% identical to SARS coronavirus. These are fish that have COVID associated with their tissues, the COVID virus, essentially. Now, these are probably not growing in the fish because they're very low levels and they're kind of fragmentary. So what we think is happening is this, and that's a little gross. I'm sorry you're eating, but yeah, there. This is Thames water over in the UK, and it's releasing what look like a bunch of little brown sausages into the Thames, I believe that is. Essentially, these are fish and they are living close to the shore. And if there is raw sewage being dumped close to the shore, there's a ton of virus that comes out in there. And I think they haven't caught it yet, but you can tell from these data sets. These are all different data sets that people recorded of just a fish that they caught in the water. They are absolutely bathing in this virus. And, if you try anything hard enough with a virus, eventually it'll maybe get through. So, hasn't happened yet, but look out for that. Now, one of these fish is special. It's the one at the bottom. It looks just regular size there because I made them all the same size. But look at this thing. The thing is, it's not the largest catfish in the world. This one is, yeah, 9.6 feet, and the largest one is 9.8 feet. So different kind of catfish, but it's big enough, right? 2.4 inches of difference. But this is one of the ones that is picking up and just getting soaked in bits of probably human waste that are loaded with coronavirus. So we're doing a biological experiment that we probably wouldn't be allowed to do in the labs, but it's happening in nature all around us, and we'll find out the results eventually. So, all right, where do these things fit? All these weird viruses. They are, oh, here, there she is. All right. They are, so first off, what do they infect? So the good news is if you look at the brown branch up here, these are all the coronaviruses that you would know. And so the SARS coronavirus, the COVID virus, is in that brown group up there. All the ones that infect land animals seem to have one origin, which is right about here. And so it's only happened once that these things have jumped from water to land. And then all these other branches that are blues and greens, the greens are amphibians that catch it while they're in the water, and everything else is a fish. The light blue are freshwater fish, the dark blue are saltwater fish, and you can see that those are all mixed up in there. So these viruses are jumping back and forth between salt and fresh all the time, and they're in all kinds of fish. That's these salmon colored things on the other side. Just really good at spreading among fish, these coronaviruses. That's worrying to people like me because it means you got a virus that's very good at jumping from one species to another. And it's doing a ton of it, and it's just out of our sight because it's happening under the water. And so I think that's kind of neat. And also maybe a little bit worrying, but yeah, we haven't seen the bad things happen yet from this. All right. So our next question that we wanted to know is, all right, now that we've got all these viruses to work with, are the viruses behaving like one genetic unit or a bunch of units? Meaning, does the whole virus evolve as like one thing, or do different parts of it evolve at different rates? Either one could be good or bad. I guess we would want it to evolve less in general, but we'll see. So the current vaccine does work great against this. This is the progress of these strains over the last year or so, and you can see the red one is very much the one that's taking over the world at the moment. So the way we're going to do this test is we're going to make a bunch of family trees. It's like, if you wanted to test if all of your kids are actually your kids, you could do this. You could sequence their genes and you could make a family tree and you could see if all of these kids cluster right next to you or they cluster next to, the milkman or yeah, person down the road. And so we've got these all color coded, and so don't pay attention to anything but the colors. You want to see the colors are grouped together and roughly in the same part of the tree. So there are this many genes in all coronaviruses. And so we're making a family tree from each one. You see a couple where it switches around a little bit, but for the most part, all of these genes are telling the same story, which means that everything is evolving as one unit. They're all evolving together, at least from the NSP3 domain to a thing called the 2' O methyltransferase. So that's about two-thirds of the genome right there. What we also found out from this is that you get more similarity between these things, or rather, you get a better read on whether or not they are evolving together when you have more data that's going in. And that just makes sense. Yeah. The more information you're feeding into your trees, the better your trees are going to get. So this is like the amount of error in your trees, and each of these colored dots is one of those little tiny trees up there. And as you put in more, this is the amount of data you're putting in. As you put in more, it just sort of flattens out right down there, basically follows a power law. And so with this, we know that if we're below zero point 0.1, that's kind of the threshold where you can't really get meaningfully better than that. There's going to be variation within that range always. So we know that if we just get somewhere around 1,500 amino acid positions in our alignment, then we should be able to get an absolutely bulletproof tree for coronaviruses. So that's a nice little thing. That lets us compare them. All right. So what about some of these viruses? Now, there are a couple of coronaviruses that were just found at a lab in China, and we've been analyzing the heck out of these. They're in red pandas, which are adorable. Look at that. And they're also in European badgers, which are pretty cute. Yeah. These viruses have made two very bizarre choices, and viruses have no actual say in this. They're not choices like you or I would make. So one thing they've done is they've taken that genome, which is normally evolving as one unit, which we showed before, and they've broken it into two pieces. I'm like, okay, that's very weird, weird way to do things. And so now this is the front half of the genome, that's the back half of the genome. And they've also gone in and they've chopped out the green thing right here, which is called the M gene. And that is a very, very weird choice because, oh yeah, so put their little names on there. The M gene encodes the M protein. It kind of looks like this. They're the black things that are up here and the white things that are over here. M has this role in making a new virus. So this is coronavirus still. First, the M is going to assemble, and the M's are going to all stick to each other. That's what's happening right into the big D right here. And then the M's are going to capture the spikes, the things that are going to stick out of the virus, and they're going to capture all the cargo that's going to go inside the virus. The M's are the only thing joining the outside to the inside of the virus, and they're the thing that lets the virus actually go out of the cell. And so deleting that is essentially suicidal. And yet, here we have one of these dumb viruses that has completely deleted it and is growing in mammals and appears to be doing just fine. We searched high and low because we kept thinking we must be missing an M gene. We built every single gene from all of these data sets, like six gigantic, multi-million base data sets, nothing in there that could even remotely be called an M gene. We assembled all the genes and then we compared them to all known genes and threw out everything that we already knew and looked at just the unknowns. Nothing remotely M-like. But what they have are lots of copies in both of these animals, pandas and badgers, of what's called an endogenous retrovirus. So it's a thing that is built just like HIV. And we know that if you take the HIV core you can actually load coronavirus proteins on the outside, and you can load coronavirus material on the inside. So what we may have going on here, we haven't proven it yet, and it'll take some work to do that, but it may be a sort of Frankenvirus where the virus got into a host that already had a bunch of another virus, parts of that around because they're embedded in its genome. And it has taken the weird step to cut out one of its own genes and just use the thing that is in plentiful supply. These things exist in humans. Our genome is about 8% copies of viruses like HIV. And so theoretically, we could get viruses like the ones that are in the panda and badger if that is how they're working. And that's also really neat and really weird and has not been seen before. Sort of a virus that's working together with another part that's already in the genome to make new virus particles. Yeah, very strange, very unprecedented. Now, the other thing that was weird about these is that usually our viruses are a monolith. They are one nice piece. And we got a bunch of viruses that are now doing this. They're essentially breaking themselves into two pieces, twice as nice, right? Like they say in Texarkana. But they're two little pieces, so maybe more like this. Yeah. So the question is, what does this mean? Why are they doing this? Because this comes with problems, and like the benefits must outweigh the costs, or else these things would not exist, essentially. So if you're just looking at the new viruses that we found, it's all the ones that are in bold here. These are all brand new ones that nobody else knows about yet. And we found a bunch more of those panda viruses after they published the first one. And so this is before our study. This is after our study. We're basically doubling the number of these weird segmented genomes and also non-segmented genomes out there. Here's a little picture of the genomes. They've got all the usual stuff, a big gray gene in front, and then there's always a spike, which is our yellow gene. That's the thing that sticks out of the virus, and an M, that's the thing that joins everything together, and an N, that's going to be your little cargo protector. And those are there in all the viruses, except the two weirdos over here that just have an S and it looks like half an N. So one of the problems with splitting up like this, splitting your genome, is that you end up with a packaging problem. And so I put a picture of, I Googled car seat, getting two kids into a car seat tantrum, because it's basically the same problem. This virus now has two pieces, and so it can grow them each separately, which is great. But unless those two pieces come together and are in the same place at the same time when they go into a new cell, you have no infection because each one is only half a virus, and half a virus is no virus whatsoever. The tips below, we have not tried these for growing viruses, but apparently they work for getting toddlers in the car. So we don't know how the virus is solving this problem. We have some guesses, but we need to find one of these viruses, probably going to be in a fish store, like a pet store. And then once we have one of those that we can grow, then we can start to ask those questions and answer them scientifically. Now, the other problem that coronaviruses have is more of an unpacking problem. So they have one long, monolith, like a big, big old genome, and they have lots of genes in there that they need to be able to get out and make copies of and make, hundreds and hundreds of copies of some of these parts compared to only one or two copies of the other. Yeah, big, big problem. And so, what we've been able to figure out so far by looking at all of the viruses and all the little pieces, occasionally there will be pieces in amongst the rest of the virus that actually don't look like the other parts of the virus. They're joining two parts of the virus together. And from these, we can go back and sort of work out how the virus is actually working, how it's building itself. And what we come up with is that, all right, for the single segment, like the normal coronaviruses, like the COVID virus, there's actually three different ways that they can work, which we thought there was only one way before this. And so as you're making copies of all the smaller genes, you get to this same sequence, and it's the same one. That's why it has the same sort of lavender dot there. And when it gets to one of these, it's going to decide, is it going to jump ship, jump up to this little spot at the front and then finish, or is it going to carry on? And some do each of those things. That's the normal case. We also found this weird case where there are actually two different sequences it can leave and join to make functional, what are called subgenomic RNAs, to do its unpacking. So it's got like two suitcases basically for just one genome. Weird, excessive, but potentially it gives the virus more control because it has a different way to make different things. And so it can make more or less of one of those things by controlling how each of those ways works. Third way we found is, yeah, unexpected, and it's still matching. There's a bit down here that matches a bit at the front, but once they match, they just stop like that. So, okay, if those are the three ways that you can make a non-segmented virus, how do you make a segmented virus? How do you unpack those? And the answers are basically the same way. So most of them have all of their packing and unpacking stuff on the one segment that needs to be unpacked, which is the second segment, which is the one that has the spike and all the things that make new virus particles. And so it's just joining to the front end of its own little mini RNA. And it's like this other RNA, just it's in the same cell, but they don't talk to each other. They just, they're just there. Yeah. And so then we are asking, well, all right, what about these weirdo viruses like our panda virus? And they just do the strangest stuff. You have multiple stopping sites for each of these. You have one segment that thinks it's a subgenomic RNA. It thinks it's basically this one. And so it's jumping up here and putting an extra part onto itself, which is insane. And then you have all these other little pieces that are reaching up there and stopping, or they're doing all of this, but not on the second segment, on the first segment. Yeah, just absolutely bananas. But yeah, that's what it's looking like right now. So there's a real broad set of possibilities for the way these viruses can work. And we didn't know about any of them until we started finding all these new coronaviruses. All right. So the next question we wanted to know is like basically, is this thing going to kill me? How bad is this? And the way we would frame that question is like, all right, what are the evolutionary rates like? If we compare the evolution of a thing that has one segment versus a thing that has two segments, are they different? Are they roughly the same? Yeah. How does this work? And so we got trilobites up there because they evolved really well for a while and then not so well after the Permian extinction. So. This is a comparison, essentially, which we can do by comparing the genes themselves. And so we're comparing the evolution of the first half of the genome versus the second half of the genome. So, okay, that's good. And here's what you find for normal coronaviruses. So these include the SARS coronavirus 2, the COVID virus. That's in one of these blue square viruses. This is just like not quite all of them, but enough of them that it covers all their genetic diversity. Um, and so you can see that, okay, so every time the main unit, so this is the unit that's copying the virus, this is the unit that is packaging up all the parts and sending them out of the cell. And it's like every time you make one step here, you're going to make two steps in the spike gene, for example. And that seems to be pretty normal. And every single one of these viruses seems to be following the law. So we can even draw lines on this and say, okay, this is how coronaviruses evolve. 95% of your data stays between the lines, and the rest of them are probably, it's the error is probably on us that these things are outside. So, all right. What happens if you have a virus that's already broken into two parts, but you need both parts to make a complete virus? How does the evolution of the front compare to the evolution of the back in this case? And I'm real proud of all these little drawings of the fish that I made. I traced all those pictures. Yeah. Here's what you get. So these little dotted lines, those are the same as the little guardrails on the previous one. So this is where coronaviruses should be, and all the gray sort of hollow circles in the back, those are the normal coronaviruses doing their normal thing. What you find is that all these colorful dots, which are all the different kinds of segmented viruses that are out there, these things sit above the line, which means they're having a lot more evolution of their structural genes compared to their non-structural genes, which means when they split the genome, this half of the genome now evolves at something like 2 to 3 times the rate of the first half. Now, the reason why you need to have a COVID shot and a flu shot and every other shot every year is because the virus is constantly changing. What we've got here is a strategy where when this virus splits itself, and there are split viruses in mammals now, split coronaviruses, it is now able to double or triple its evolutionary rate. And so that's bad for us and great for the virus, apparently. All right. Oh, that was a lot, wasn't it? So let's talk about quantum optics, which is basically shooting lasers at things, and we can use the lasers to kind of read out what's inside. And we're going to do stuff to viruses here. It's still going to be connected, but yeah, with cool little laser setups. Yeah. All right. So what are we playing with? We are messing with what's called the main protease. So the main protease is one of these unpacking proteins. So the big gray bit here comes out as one long chunk. And what the main protease of the virus does, the virus makes its own thing, and it's like a little helper that comes along and breaks the chunk into little pieces. And then each piece can go off and do its job, and they can fold up into, big, awesome complexes. Yeah, they can be together because they want to, not because they have to. And so, yeah, the inhibitors like Paxlovid, which may be the only coronavirus inhibitor that you're familiar with, those are going to inhibit this thing. So they just stop the virus from unpacking its big gray protein into the little bits. And when you stop it, the little bits do not work unless they are unpacked. So that's kind of cool way to stop the virus. So that protease, the main protease, is the thing we are messing with, and we are looking at it to try and figure out, all right, how do you make a good antiviral? Like, what are the characteristics? How do you make this thing work? And can we tell the difference between a good one and a bad one and predict what will be a good one using lasers, essentially? So what we found is that inside the protein, there are these three little rings, H163, 172, and F140. So these are just the names of particular amino acids. They're kind of buried in the core of this protein. And what those do when the virus is, or rather, when the protein is just one copy of the protein. So, like, here's one copy I'm circling right there. That copy, the monomer, is not a very good protease. It's not very good at doing its job. But when you get two of these together, all three of these things will rotate around and they will lock together. They'll do a thing called pi stacking. Yeah, makes a little. That's and it's actually pi orbitals that are stacking together, and it's electrons forming a little thing. It's kind of like a hydrogen bond, but it's with pi electrons. Yeah. And that seems to be a real marker for what makes the virus tick. Because when it does this, then all of a sudden the virus becomes very good as a cutting protein. So this enzyme is good at its job when it's dimerized and when these three things lock together. So yeah, pi bond. There we go. Yeah. So, what is this pi bonding thing? Well, so what we can detect is benzene ring breathing. So here's a benzene ring, just six carbons right in there. And what happens to this, well, my little animation didn't work, is that when you hit it with a laser, it's going to expand and contract like that. And that motion is going to eat up a certain amount of the energy of the laser. And so the light that comes out is going to be shifted down by exactly this amount of energy. So you'll still get a photon of light out, but it's going to end up being a different color, essentially, because the vibration energy that it had to put in ended up, yeah, absorbing some of the energy from the photon. So what you want to look at on here is here's the monomer protein. And so we can get this nice little spectrum. That's all these little bumps and peaks. And that's just what it looks like when you shoot an infrared laser at these. Everywhere that you get a peak is where we're getting some basically vibration that's eating up some of the energy. And as you put in more of the protease, it's going to turn into dimers. And you can look at the red line and the blue line and say those are pretty similar. But there's areas where they're a little different. And you can actually subtract the two lines from each other, and then those areas just jump out. And there, it's right there. See that? Like a little heartbeat. Yeah, on a monitor in the center. That's the signal. And the thing that is right there is all these rings. So these rings breathing, those give off a signal at about 1003 wave numbers. Yeah. And so that's what happens when it dimerizes. And so we want to see, all right, what happens when you have put in good inhibitors versus bad inhibitors? And so this is just the subtracted version. So this is just showing the difference between those two maps. But look at that. The black one and the orange one, these are the really good inhibitors. This is actually the active ingredient in Paxlovid. This is a slightly better version that we made. And you get massive little changes right there. And they get sort of dampened out as you put in more of these things. They just absolutely suppress any vibration in this area when you put in lots of drug. Very cool. Whereas when you take inhibitors that are not very good, like not binding strongly enough to be useful for human use, you get a little tiny bit of that, but it's much less. And it absolutely correlates with how good these things are at inhibiting the virus. So it looks as though this change, this three amino acids locking together, it's kind of like the little, safety cap on here. It's just like a little indicator. When that happens, that makes it first a better protease, but second, all the drugs seem to cause the same change. And the ones that cause this change even harder actually turn out to be the really good drugs. So that's the thing we should be focusing on to actually drive making better versions of drugs like Paxlovid. Yeah. And we got a couple of these, and they seem to be pretty good. But now we know what to look for, and we ought to be able to do this much better. Yeah. All right. So next thing I want to go, and so this is we're like two-thirds of the way through. We're doing great. Yeah. So I want to look at some virus interaction dynamics. So when you get two viruses together, like one person has one virus, one person has another, what happens if you're in the room and you get both of those viruses at the same time? Are they going to make it worse for you? Are they going to fight each other so it's not so bad? We want to know. And what are they going to do out in the real world? Because all these things are circulating amongst humans all the time, and deer and various other animals now, maybe fish someday. Yeah, how's it work? So what we've got are two viruses. Now, the one that I've colored green here, same virus, same COVID virus, SARS-CoV-2. But when this goes into cells, it's going to turn them green. It can make a fluorescent protein that goes green. This is the normal virus. It's just a strain that we isolated from somebody in the lab. And this thing does not glow green when it infects the cells. It just kills the cells like a regular virus should. Yeah. So what happens when you throw these two things into the same batch of cells and you let them fight it out? Do you get more fluorescence? Do you get less fluorescence? Does one of these things win? Do they help each other? Do they cannibalize each other? What happens? Yeah. So here you go. So this is this blue line here. This is how much fluorescence you would get out of the green virus if it was all by itself, nobody else around. This one in green is how much you get if you have the same amount of fluorescent virus and non-fluorescent virus. There's plenty of cells to go around, but the two of those things just hate each other. They destroy each other. And so they compete. They go straight into competition. And you can try it with different amounts, even if you lower it so there's one of these competitor viruses for every 1,000 of the green viruses, you still get a big dent. It's way lower than the amount of green you would get out of just the green virus. So yeah, there's a winner and there's a loser. Basically, the normal virus beats the pants off of our green virus over and over again, and they just compete. They don't help each other. So maybe that's good. But wait, there's more. Yeah. So we wanted to see, what if we have one of these viruses and it has resistance? That's the Rebel Alliance insignia on there. And so it has resistance to one of these drugs. So we have some non-commercial drugs, like things that we can make it resistant to, and then we can check and we can put that drug in there. And so basically, this virus is the only one that should be able to grow. This virus will basically die unless it cooperates. We're trying to see if life is on the line, virus life, whatever that is, will these things work together or will they just, yeah, destroy each other and go down, fighting? Yeah. Middle fingers blazing, basically. So here you go. So this is the amount of the green virus that you get when you're treating with drug, because the green virus is not resistant to drug. And so this is really low compared to the amount you would get if you had no drug there. That's way off the top of the scale, but we don't care about that. And so what we want to see is, all right, if we throw in a little bit of this other virus that is resistant and absolutely could cooperate, will it cooperate with our green virus? And the answer is no. You can put in the tiniest little bits of the drug-resistant virus, and instead of working with the Venus virus, it just crushes the Venus virus. Absolutely. And when you put in more of the drug resistant virus, the amount of green just goes down to zero. It is an absolute knockout victory. Same thing as before. No cooperation whatsoever, which is probably good for us, but it also means that whenever two viruses compete, whichever one is like worse for us, better at growing, is going to be the one that just dominates and wipes out the other one, which means that we're going to need to keep updating that vaccine and keep getting vaccinations, unfortunately. So then we thought, well, what if we used one of the drugs that's used to treat this virus? This is molnupiravir, and molnupiravir has this neat property that it can do this. It can sort of switch. So the OH is on this side, and then it'll switch to this side. It looks kind of like a nucleotide, but it's got that little switchy part at the top. And so when it's in this mode, it's great at binding to. It looks like a U nucleotide, and so it's going to bind to A. Yeah. And when it's in this mode over here, it looks like a C nucleotide. And so it's going to bind to G, which means that if you put a molnupiravir in, it's just going to keep going switchy, switchy, switchy back and forth. And sometimes you're going to put in a G to match it, and sometimes you're going to put in an A to match it. And that is a mutation. That means we're forcing the virus to mutate more than it would like to. Because even viruses have a limit. They don't want to mutate too much or else they go extinct. So we put these in here and we tried to make groups of viruses that are broader. So you get like a whole swarm, a population of viruses. We call it quasi-species. And so we're trying to compare the quasi-species with the green one. And we made several different quasi-species. Some we made with just a little bit of drug, and we checked the sequence of all these things, and they've only changed a little bit. They're only a little bit more diverse. Some of these other things are super diverse. And yeah, so we want to see, all right, what happens? Yeah. Do these things behave any differently when they're together? And from the first graph, so here we're just looking at the total amount of viruses that come out without saying which one is which. And the total amount of viruses that come out stays pretty stable. This is just the green virus on its own. This is just the competitor virus on its own, if you like. And if you add these two things together, they're on a log scale. You end up with right about here, which is where every single one of these is. So it looks as though these two things are now no longer fighting. They're just sort of at a détente, yeah, at an equilibrium. But yeah, if you look at fluorescence, you can really see what's going on because here's the amount of green virus you would get if there was only green virus there. And so here is the basically not mutated version of that other virus. And then here are groups of those virus that have a broader and broader quasi-species, like they're more diverse in each group. And you see there's actually a peak up here. When you treat it with a certain amount of this drug, around 4.5 micromolar, you get a really broad population. And that broad population ends up helping out the green virus. And so you make twice as much green there. It's making it go twice as hard, as fast. Yeah. So broad populations of these viruses appear to cooperate, whereas narrow populations of these viruses appear to compete. And in the lab, we're always just playing with narrow populations. You're supposed to keep things pure and, clean and everything. But in the real world's messy. And so some of this is probably happening there, which is kind of a neat thing. So this is from Deshna, and that previous slide was from Tamina. These are a couple of my PhD students. Yeah. So yeah, not competition. It's love at first sight. Yeah. Between these little viruses. It's a Christmas miracle. They're helping each other out a lot, but it's dependent on genetic diversity within the virus swarm. Yeah. All right. So That's bad for us. But can we use this genetic diversity against the virus? Because that's what you've always got to be thinking as a virologist. How can I bully this virus harder? Basically, that's the job. Yeah. And so viruses actually they do better when they're making mutations. It's like risky behavior, for a virus essentially. They're much better when they're making mutations than when they're making no mutations. But there is a limit. If they go beyond the limit, we call this the error threshold, then, so like polio virus is pretty good at three viruses or three mutations or four mutations per genome. When the cloud is that broad, everything works really well. When you go over that, you end up with essentially dead viruses. And these do not work. And yeah, viruses that are five and above are almost all just completely non-functional. So, can we take a virus, which normally starts out here and then mutates up to here? The virus doesn't know, anything has changed, but can we deal with it? So imagine you had to deal with some annoying kids. You could try to placate them. You could, give them ice cream, something like that, and that's fine. That's like not vaccinating, essentially. You say, "Well, let it run." Yeah, but in the end, is that actually going to work? I would say no. Yeah. So there's this other strategy, and this is the part that's not from my lab. This is friend Volker Thiel over in Germany. And so what they're doing is they're taking the genetic code and they're trying to make it a little closer to failure and a little farther from safety. So if you want to make the amino acid leucine, so here's your genetic code possibilities. You got six ways to do it. Three of them, UUA, UUG, or rather two of them, are very close to three of the stop codons. So UAA, UAG, and UGA. Just make one change, and then we're one away from a stop. So they call these one to stop. And then these other things over here, they're actually two away from a stop codon. And so they call these no stop, but it's really two mutations would be required to stop. The virus can only mutate, and it mutates at a very fixed rate. It's been selected by evolution. And so if you take a virus that has to change, that's got this change built into it, the question is what does that do to the virus? So you can take a virus that's got a mixture of these one to stop codons and no stop codons, and they're all just coding for serine or leucine. But you can change it so that they are all one step away, one mutation away from a stop codon, and a stop codon means a dead virus. Or you can take it so that they're all two mutations away, so they'd have to make two changes at the same spot to get to a stop codon. So these are, kind of bulletproof viruses. And so what does that do? And can we actually use this for good? Essentially the strategy is this. You've got this annoying problem. And so imagine they're viruses, not kids. What we're trying to do is this. We're trying to put them real close to the edge because we know they're going to change. And in this case, when the virus changes, it's going to fall off. And I don't have a good falling off animation, but you can imagine. I don't need to traumatize you over lunch. Yeah. So this is the one to stop virus, and this is it's going through testing over in Germany at the moment. It's a live attenuated, so like a live version of SARS-CoV-2 that you squirt up somebody's nose, and when you put this thing in, the one to stop viruses actually they don't kill the mice. So this is like the mouse weight loss plan. If you put in a virus that makes them sick, this is like the normal virus. Their weight will go way down because they don't feel like eating anything when they're sick. But if you put in these one to stop viruses, the mice just continue to grow almost as normal. It's the same virus. It encodes for all the same parts, but each of the parts is fine-tuned that little bit closer to failure so that when the virus slips up, boom, it goes straight over the cliff. No recourse, no way out. And what you get is a virus that grows pretty well in the nose. This is the nose amounts. Doesn't really grow anywhere else in the body and generates really strong protective immunity. So this may actually be an answer to making a possibly potentially better vaccine. You can't take it yet. It's not approved for humans, and I don't know if there's enough money floating around that it'll ever get approved for humans, but it's a pretty good idea. And I think you'll see this coming back for some virus at some point. So there you go. Your Christmas stocking is overflowed with viruses. I'm happy to take any questions you have. That's all I've got for you for today.
Thanks, Newman. Always cheerful. I appreciate that. The stocking helps. So we'll give you a little round of applause here in the room. Thank you. Hold on. And yeah, we'll open it up for questions. I know TC folks are also going to be taking a tour, so we won't keep you too long. But anyone want to ask any questions? You've already answered most of mine, which were, what do we do with this stuff? Because the science is largely above my pay grade. But yeah, anyone jump in on
Yeah.
that?
And what we do with it is we take it and like it, essentially, when it comes to
Exactly, yeah.
viruses. But yeah.
Pretty cool. Any questions before we release Dr. Newman? Anyone of our Zoom friends have any questions? That's it. Okay. Well, you've fulfilled your contractual obligations another year, Dr. Newman. I appreciate it. Now, thank you very much. I will get this put up on our YouTube channel for those of you who want to watch it again in the future. And we appreciate it as always, man. Enjoy College Station. So, yeah. And apparently Hawaii, which is your Christmas experience right there.
Will do. And for any of the honor students, TC or Tamit out there, I've got a grad student who's putting together undergraduates to help build some of these viruses. We've got tons of these things that need to be assembled, and we're trying to work on coral viruses at the moment. If you want to do something like that, ask Dr. Nakashian, drop me an email, and yeah, it's not a thing you would have to be here to do. It's very remote, work at your own pace. And I don't know, you might do some good for the world. So yeah, the offer's there if you want. Yeah.
Oh, thank you, sir. I know I can think of one who's actually ironically not here. He's working today, but yeah, there's a couple I can think of that might be interesting. But all right, man, I appreciate it. I'm gonna go ahead and stop the recording now.